Comparative modeling is today the topic, and again there will be, there will be no lecture on Tuesday the 3rd. So actually there are a couple of Thursdays that will be skipped. Ascension, Whitsun, and Tuesday the 3rd. So some of the slides that I present today are from Andrea. Let's repeat the issue of comparative modeling. The first thing is, in principle, we know from Epstein and Anfinsen, we take a protein chain, we throw it into a solvent like water, and it adopts a unique three-dimensional structure. Everything else is determined by the physics of the interaction between the amino acids on the one end and the solvent on the other end. Is that statement true?
And there are things like chaperones that kind of influence the folding of proteins.
So let's begin with the first statement. So Epstein and Anfinsen showed this experimentally, that there are proteins that behave like that. Is that still true? So that's 50 years ago. Is what you say invalidating that statement?
Is it, but they did it without chaperones and everything, right?
That is typically done in a tube, and in a tube you typically don't have chaperones. In fact, chaperone folding or refolding through chaperones is something that for which you need an in vivo system, or something like an in vivo system.
I think there are some proteins which have also different folds, which can fold in different ways.
That is also correct what you say. So again, not all proteins. So if I looked at all the pairs of proteins that have 100% sequence identity, then they would not all be identical. And it is true that some proteins have a tendency to adopt slightly different structures. Porine is an example, where in fact in the porine example, you know that it's a typically mutation that moves it from alpha to beta, but it's a very, very big change. And we do know that in Alzheimer we have an induction of a structural change. We have an example of that one. So that brings me back to that question. Does that invalidate that experiment? Is it true that they saw something in the tube then that you would not observe today? It's not true. That is correct.
I think you would observe it today, right? But
yes.
Not in all cases. There are exceptions to the rule.
Yes. Now that brings me to the next question, of course. I love your statement. There are exceptions to the rule. So is that the rule?
No, I think they had really good luck that they choose a protein which,
I would say this is the rule. If you throw in one protein and it adopts a structure and you do it again and you put it in the same liquid, it's going to be the same.
Be careful. What you say is true, but let's just keep that out. So one thing is absolutely clear. We assume that the porine or the Alzheimer, these kinds of examples, are the exception.
Yeah.
So that we absolutely don't believe is the rule. Let's not discuss that issue. So the question is not, does it do the same thing again? But the question really is a little bit more along the lines of what Dennis says. Your name is Dennis, right? Dennis, the one I don't have on my pictures. And so what Dennis says is we need some help. So back again, if there were to be some cases that need help, and those that do it themselves, the question is not those that do it themselves, do they do it again and again the same way? But the question is the ones that do it themselves, the ones that need help, of which we have more. So are these in fact the chaperone needing ones the exception, is the question. Have you read anything that would answer that question?
I know that normally when a protein is translated, there are already a lot of proteins binding to it, and so it's never really alone in the solution. So
that is correct. But now let's get back. That is in fact an important observation, because I believe we all in this room admit, or it's all clear to all of us, that there are proteins that behave such that this is true. You put them in a solution and they adopt this the three-dimensional fold that we know they have in the cell. We assume they have in the cell, or in some cases we know. For those cases, the kinds of interactions that you talk about happen too. Although we can reproduce in vitro that is not needed. So the very fact that we observe there is an immediate, let me misuse the term, chaperoning already there, sometimes really is chaperone proteins and sometimes not. So I'm misusing the word chaperoning here. But there's an immediate binding, an immediate guidance, an immediate environment of other proteins that may help. Is not necessarily disproving this. Okay? It could be like that for most proteins. That in fact there is some interaction locally, directly upon each, when one amino acid comes out, or every trimer or whatever, and then there is some interaction with them, and then the structure is formed, but it would do the same in solution. Okay. So but that is an important point, because in fact we more and more there's evidence that the DNA sequence is relevant for the folding. That now is only explainable if all of these interacting partners that you talk about would have an active role in folding. So that much we know. Oh no, it's not known. But this is something that is just over the last years. Let's imagine just a boiling water pot, and it's sort of boiling over. So there's something under the lid that seems to be happening. And over the next years, we're going to see some publications that will produce, I'm predicting now, will generate a lot of surprise. And most likely that will be the case. What we in fact, so I asked you whether that is the exception or not, we still don't quite know. We have observed ever since over the last 50 years that chaperones play a more important role. So with the discovery of chaperones, their importance, or our degree to understanding their importance, has increased every year. Over the first, so the chaperone field started in the 80s. Now it got bigger and much bigger in the 90s. Initially, those were all, most of them were heat shock proteins. So in the background of, we have some problem. Heat shock, so you have put heat cell under stress, and the cell reacts to the stress because the stress leads to, what does it do, the stress, the heat stress in a cell?
Denaturates proteins, right?
Exactly. So you unfold proteins, just the same thing that you do when you boil your egg. That happens literally, it sounds funny, but it's scary. Because that exactly is happening. And that is typically what we have chaperones for, heat shock proteins for. And in fact, that is explaining why the most overabundantly expressed proteins are heat shock proteins, are chaperones. That's what we believe today. We still don't in fact quite know at this point whether that is the exception or the rule. What we continue to believe is, however, that if I started from the right domain, the right folding block, which is not the protein at all, but from the right domain, then this would be the rule. That's what we continue to believe. And there is evidence for that. But, okay, this is the background. Now, still, this background tells you a lot about the importance of the underlying biophysical principles, about the degree to which we should be able to, in fact, infer structure from sequence. And that, again, brings us back to the topic of today, the comparative modeling. This is KRAS, an oncogen, the structure of RAS. And here is RAS with a point mutant, the point mutant being in red, the rainbow structure being RAS. This point mutant, the glycine, so G12C means the glycine at position 12 is mutated to cysteine. When you look at this, there is no dramatic change. In this particular case, so you can quote that, I show it to you as an example, for I can predict structure. I have a single amino acid change, no problem to predict the structure. In this particular case, what the story is hiding here is this, in fact, is a somatic change, so it's something that happens in people. And so RAS is an oncogen related to cancer. There's a therapy that targets KRAS, and the therapy becomes inefficient for the ones who have that mutation. So the drug no longer works. So this tiny change here in the way the GDP is bound, this tiny change matters for the efficiency of the drug. Still, the message here is you have a mutation, you keep the same structure. You have now, in this case, we look at two related molecules, KRAS and RAS in human, 85% sequence identity, essentially still the same. We go to the next level, down to fly, 28%, and still essentially the same structure, right? Again, this is on the level of 28% sequence identity. It's very, very diverged already. Fly, human, clearly not the same organisms. Still, overall, not that dramatically different. Now we go to the next level here. That is the level of the point where we lose the function, but we still stay in the enzyme, the hydroxylase, in this particular case from a bacterium in purple. And again, you see that it's essentially very, very similar. This shows the evolutionary pie chart, bacteria, eukaryotes, so RAS is a huge family. Since that is the case, we should be able to do comparative modeling. Comparative modeling essentially uses that kind of idea to predict structure for proteins for which we don't know them. Let's put that into perspective. So when we use structure to predict function, essentially we assume that it is a particular aspect of the 3D structure, so the way a histidine triad is spaced, the three histidines are spaced, that's the way you can bind. That's why the structure is important. And essentially for this histidine triad, what is around it doesn't really matter that much, right? To some extent, and I'll show you in a minute, downside to that. So this method here, Conserve, from the lab of Nir Bental in Tel Aviv University, essentially maps out structure, surface, and conservation. And conserved is dark purple here, not conserved is this dark blue or light dark blue, ocean type of colored. And you see that the sort of more conserved surface things are the ones that bind the substrate. So that is another thing that you can immediately learn from structures. You can map structures to surfaces. That's great. So one of the reasons why, therefore, we want to build comparative models is to learn about the surface. Now here's an example where we have two proteins here, and these two proteins in fact have different electrostatic surfaces, although one is a structure and one is a model. So the model is built on the other one, but still has very different surface properties. This is an example what you can achieve with comparative modeling. I wanted to put that in the background behind what I'm going to say now. Here's another example where essentially the fly is interpreting the talk. I don't know. Okay, let's just keep the fly happy. So we have some elements here, and the remaining part of this picture is essentially done by comparative modeling. Again, the background simply is you have a bunch of structures. You start with a sequence of unknown structure, and you want to match that sequence to that known structure. Let's begin with the numbers of sort of structures, and the numbers of, there's a typo. This is 55, I'm sorry. And I copied, this typo will follow me throughout the slides here. It's 55 million proteins in UniProt at the moment, about 400,000 in SwissProt, and about 100,000 in the PDB. That's sort of the coverage of the experimental structures. The way that evolves over time, so a year ago, this was in fact 35 or 34,000. 55 again is the real number. A year ago was 34. UniProt was this, and PDB was that. UniProt indeed is smaller now because UniProt has moved into deep annotation. So rather than having as many proteins annotated as possible, they try to improve the annotations as much as they can and remove records if they're not good enough. Now let's get back into analyzing the uniqueness of the PDB. So all PDB today, then it was 70,000, today about 100. Just looking at the number of pairs that have 100% identity, you see drops it from 70 to 42. So again, we have about 28,000 structures that are identical in terms of their sequence to something that is already in the PDB. Why?
If they have different folds, or if they have different conformation bound with the ligand.
So the first one is the story that you were saying, and in fact it is true. So we can induce different structures, and it is true that some proteins adopt different folds, but this is the exception. So the different fold story is not really explained at the second explanation that you have. That in fact is the important one, Verena. So the explanation that we have in fact the bound with a ligand or not, the ADP KRAS that I had before and the KRAS without it. Or in the example before, I have the KRAS, no, well, the point mutant is not 100%. The point mutant would be sort of this part. Any other reason? Nico?
Maybe some proteins are just present many times because the experiment has been done.
Why? That is correct, but we are asking why. Yes?
Because sometimes it's simpler for some proteins to do the experiments, and maybe they are X-ray or NMR.
That is another reason. So some of those are in fact done by different experimental methods. I'll briefly get back to the issue of how we do experimental methods. Could be NMR and X-ray. True.
Yeah, I want to say kind of the same thing. If you just with improved resolution, so they're done again because the previous model wasn't really good enough for whatever people were trying to do.
And that in fact is an example where I want to show a figure that I've seen first, and I actually should get it from him, from Sean O'Donoghue, who is now in Sydney. And what Sean shows is somehow an axis that reflects the impact factor of a publication versus the strong audience, versus the RMSD, okay? And you sort of see a clear relation. It's actually the resolution, right? So better journals publish better papers, right? Of course, you guys know that RMSD in that direction is bad. So higher is a negative correlation. So the more impact the journal, the less accurate the structure. And to some extent, that is exactly the line of an argument that you started. So you get into Nature because you're the first to do a certain structure. And then you do the structure right a few years later. Now you know much, much more. You had a lot of time to do the fine-grained analysis. You get to a lower impact journal, but you do a better job. And that is another reason. Yes, you're totally right. So mistakes are corrected. That typically takes time. And in fact, so in that sense, this is by no means easier to do it again, but you need a lot of time to do it better. And another point is really the one that implicitly you said, because it may be easier to do and there may be still some way in which you can sell it to another journal. And the other thing is, it's remarkably often really that people pursue different labs pursue the same structure in parallel. And one comes first, and the other one still will be published. Overall, this is this large figure, and somehow the same story goes on here. So 28, now we are at a point that is less than half of the 70, and we are filtering out 95%, which really is essentially actually the same story still. We have mutants, we try to see the influence, which we do for functional studies. It's still a clearly related issue. And then we actually begin to have a sort of more flat image. Again, comparative modeling is about mapping the structure through finding the related protein, and when we do the related protein, the safe zone, so where we do sequence comparison, pairwise comparisons, sequence profile comparisons, how much do you believe, roughly, we can add to the experimental structures that we have by this sort of simple alignment tool? Let's call it BLAST and sort of psi-blast. Not full psi-blast, but sort of without psi-blast, blast, without the eye. So the first sequence to profile kind of methods. How far can we cover it? So far, so we are standing at 13% roughly. How much is that going to add? As much? More? I asked 2 questions and you say yes. Tired.
Yeah, you asked how much and then much more, yes.
Much more. Oh, much more was the
How
part.
much?
So much more. A factor of 2 is certainly by all means much more. Again, this is wrong here. This is roughly what we can do. Now, again, I showed you the length versus percentage sequence identity. Blue, the same structures. Red, different structures. I said everything above, essentially the gray curve, the HSP curve, has a similar structure, but there are tons of things down there. This is the midnight zone, so going further, beginning where we can no longer get here, where we may really need profile, where we fight against the background, true signal versus bad signal. That is where we're going in. Just to repeat the numbers, this is where the RAS and RAP6 for human and fly were. This is where the KRAS that I showed you and the bacterial protein were. So in this case, in the realm where I showed you the bacterial structure, and you remember the purple, and maybe I should bring it up again, how similar the purple was to the others, right? But that's a place where, in fact, less than 1 in 20 of the hits you find is right. So we are deep in the twilight zone, beginning to see the midnight zone there. What can we get there? We add something like that. Another sort of 20 percentage points. Beyond that, in this midnight zone, again, all of you, this figure is clear? Do you all remember that? So that's the midnight zone. Essentially, what it plots is the distribution of pairs of proteins that have similar structures. The statement is that the vast majority of structures, of similar structures, of pairs of proteins that have similar structures, are at a very, very low level of sequence identity. So have no recognizable level of sequence identity. So are way below the purple and the green that I showed you before. Okay? That's the entire database here. We cannot reach there. What we can do in this realm here really about structure is we can predict secondary structure and solvent stability, in some cases contacts. That's what the remaining part of the lecture will be about. That's this pi. Now, this is essentially modeled on some old data. I said it was modeled on the 99 data. I didn't say that, I only put it on the slide. That slide was done by running Swissplot. So originally we did a dynamic programming against Swissplot. We couldn't do a dynamic programming against the whole UniProt because we didn't have the CPU for that. We still can't do that. And we had a lot of knowledge about Swissplot. That's why this was all based on the Swissplot estimate. What if I had used UniProt? What if I had used the entire proteome? Would the numbers change? Because Swissplot is again biased. Similar reasons as we talked about before. We have the same protein again with a new experiment, with a new or slightly altered protein version, slightly different organism, tried it in mouse and rat, just to, or fly, that type of fly, just to see changes. Would that look different for this pi? So again, the pi shows the reach to which we can apply easily, or more or less easily comparative modeling. That's the Swissplot view. That's the genome view. Very, very different. So the numbers I showed you before are optimistic. The good news is this: if I just wait, I sit down and wait, things evolve in a good direction. So this is sort of the pitch talk to study bioinformatics, right? You begin to develop a method in 1991 that does comparative modeling, and then you sit down and drink coffee, right? And then you look again 10 years later, and everything is much better. Isn't that great? And I can tell you, if you work on it, the improvement from here to there is immense. You will never reach that. So you better wait it out. This is from, I'm going to show you 2 slides from Jinfeng Liu, who is now at Genentech, or now since 2007 at Genentech. And one reason why I show this slide to you, because he's a smiling father now. But those were computers in the time. He did a PhD with 16 publications. Beat that. Go out there, beat him. One of the publications, or two of the publications, are here. Blue, number of open reading frames or protein analogs. Human is still not complete at that, still an estimate, incomplete estimate here at that point. Blue, genes for eukaryotes, prokaryotes, and archaea. Red, number of things for which we have a hit in the PDB. So this is roughly what you saw before about the whole genome view, but a little bit significantly worse, because essentially we are asking how many do we really have something almost directly in the PDB. If we extend it a little bit, so with a PsyBlast, then these are the numbers. So ballpark 30% we can do by comparative modeling. That is 30% of the proteins. I find some similarity. That does not mean 30% of the residues. Now, let's get into methods. First, lingo. I talk about comparative modeling. You may have heard homology modeling. What's the difference between the two?
Yeah, should be I don't know, but comparative is probably sequence identity or something. Mm-hmm. And homology is, yeah, homologous genes are ones having a common ancestor. So.
Verena, you just graduated from the School of Biology. So biologists are about observing effect and finding a reason for it. Well done. It's not true, but you know, this is exactly the biological thinking. This is great. I'm really impressed. In this particular case, really, this is more about past and present, let's call it that way. Or some people, other people. Historically, it was called homology modeling because, now let's get back into why do similar sequences have similar structures. And again, if you randomly created sequences, this is not the case. So you would create a lot of things that we at least don't observe to have the same structures. They may not have fold at all. That is our current explanation. But that implies that the reason why similar sequences have similar structures is because there is an evolutionary history. There's a functional reason. We have evolved to having a similar structure because we needed a similar function, because they are related. And that is sort of all captured by the word homology. And that is why it was called homology modeling. Then it was found out that we in fact now get much deeper in terms of divergence of sequence. We're no longer sure, but there is an evolutionary relation between these proteins. We pick up things where we no longer quite know. In the Rost case I showed you before, we absolutely believe that that is an evolutionary relation, so that's a homolog. But not it no longer, at least it no longer has the same function. And we certainly go deeper where this story becomes unsure, so some people call this comparative modeling now. Target and template. Target is the protein to model, template is the protein we model from. So the target in this particular example is here, and then in this particular example there would be two templates. And again, so we have to find all of the related ones. So the first one is identify the template in the database search, one or many. And the next one is to in fact align target and template. And the next one then would be to build a model. Once we have a model, we can assess it. Assess means now, in fact, I have a new sequence in that 3D structure. And the new sequence may imply some clashes of side chains. So there's just not enough space to put it the way I have it in the protein sequences that I actually have, because the side chains are longer, right? Or it could be that suddenly in my new structure I have two positive charges next to each other, which I know is energetically absolutely unfavorable. So maybe I have to sort of change that, and that is in fact refinement. Refinement essentially means, can I move things around? Without, the problem with that is, so this is the kind of thing that I absolutely cannot show with my fingers. Just imagine that you have this protein chain that goes through and through and through, and then you want to change one point inside. There's high density inside. You have a lot of connections, so moving one thing around makes the next thing move around. So the refinement is not that easy, because everything you do inside of a protein structure is surrounded by interactions. So meaning one change implies a lot of changes, and so what you do good for one residue may be bad for another, and that is immediately a challenge for refinement. Up here in this region, again, we mostly use sequence comparisons. Down here in this region, where we get far away in terms of sequence, we begin to use structure for the refinement, and I'm going to show you examples of that. So the identification of the template is typically done by PsyBlast, HH Blitz, threading. The template is typically aligned. I'll show you, I'll continue on this in a moment. Before I do, let's just look at the challenges for finding the template. So up here, so when target and template are very, very similar, essentially everything is easy. Finding the target, doing the alignment, and the problem really is to do the model right and in fact do the refinement and do all these, so care for the details. So ultimately that leads to a situation where the speed of modeling becomes an issue, because you want to try to see as much as possible and try to see as much as possible ultimately gets into molecular dynamics and things like that, and that you cannot do. You're familiar with the term molecular dynamics? I take it as a yes. The lack of yes or no I take as a yes. So molecular dynamics, can you do molecular dynamics on a stretched out sequence? Let's go a step back here. Protein structure prediction. Can I I showed you the Anfinsen experiment. The Anfinsen experiment where I take a protein sequence, I throw it into the solvent, it adopts a unique three-dimensional structure, and it does that without any chaperone help. Now, let's look at the set of proteins for which that is true. We discussed before that this may not be true for all proteins, may not even be true for most proteins, but the subset of proteins for which that is true. Can I do that in a computer? One at a time. So there was there? I didn't hear. I didn't locate. Where did it come from?
I said generally not.
Ah, you were the one. Totally wrong.
I had a lecture last year, and they showed. I think they showed that I did it with a short protein, which they heated, denaturated. I think simulated the denaturation, and then they simulated the full.
What is a short protein? Are you talking about vimentin 30 residues, or?
Yeah. Was something like that. Yeah.
So you're talking about David Shaw. Yeah.
Professor Fishman showed it.
Yeah.
Okay.
Yeah, Professor Antes showed a video of it.
Do you know what is behind? So this is a science publication of David Shaw.
Yeah, I'm not sure if it was
Anton. Anybody knows what Anton is? That's the machine. So David Shaw built a computer. Big computer. That big computer is Anton. And anybody knows anything about David Shaw? So it's a big computer. Not only a big computer, but it's a special purpose computer. It's a thing that essentially only does molecular dynamics. It's optimized. So everything, this is totally enslaved to the topic of folding proteins. Special purpose machine that is completely built with the potentials and the task in mind. And every aspect of it is optimized, including the potentials are optimized to fit to the computer. So this is on a scale optimally fit that I don't believe has many other examples in the world. This way they can do way more than any other group in the world. Group is the wrong word here. It really is more an institute than a group. But so the first publication of that was the vimentin, so 30 residues. They can do a little bit more today, but not much more. So for what is an average protein length? A typical protein length?
300.
So 100 is another example. So many domains are about 100 residues long. So vimentin is unusually short. So nobody at this point can do that on a full-length protein. So what typically is done with molecular dynamics is we have a structure that is close to the real structure. And that comes, for instance, from an NMR, or it comes from X-ray crystallography and is refined. So now we are in an environment of the right solution. And all we do is we wiggle a little bit around to avoid bad clashes. If we did that, so we start from not a model, from a real structure, and we now want to, for instance, see how, in fact, is I showed you in the KRAS example, the glycine 12 to cysteine mutation. So now I can model that mutation with MD. How long would that take? Not that long. That is correct. It's not long on the scale of paying $50 million for a machine. Or no, there's certainly more than $50 million in this. But on that scale, it's not that long. But what do you mean by that?
I think we did it in the lab course at Professor Antes.
It takes only a few minutes.
Yeah, but we have the longest separation.
But that is, a few minutes is just one solution. There's one optimization. This is not going through the space of possible.
No.
That's not sampling the potential space. So if you really search the potential spaces, so now we are talking about something, let's get back into the problem that in our group we have approached, or Mark Hoffman in our group has approached, which was we have a tool that predicts the effect of single mutations, single amino acid mutations upon function. Let's just take the subset, and for those we have 50,000 of these mutations for which we know their effect. Yes, it's neutral, or no, it has an effect. And just take these 50,000, map them to the structures for the cases where actually I really have a structure I'm very close in comparative modeling, which I believe left us with 10,000, and now run MD and see what's the effect on an MD simulation. What would that cost?
With 10,000 people.
So the number of proteins in that case was in the ballpark of 1,000. Yes. So now we are really talking about all the available time that we have on the LRZ for ourselves. It's not the entire machine, but this would be on the entire machine quite a long time. So that is still a major effort, that sort of thing, which is not that different from what you look at, because we start from a structure and we wiggle around, but we need to explore the space. That makes it much longer than the example that you quote. Anyway, so we're beginning to see here what I meant by speed of modeling. When you get further down, the quality of the model becomes the issue. Why is that? So we're back to comparative modeling here.
Well, in the level of 50 to 75, you get a very good idea of what the backbone of the protein looks like and where the secondary structures will be. But you do have to take special care of the loops.
Well, loops is part of the issue here. Side chains is another part of the issue. So they are simply beginning to see changes that are not easy to compensate for. That's as simple as that. Below here, the alignment accuracy becomes an issue. And again, this is an overall totally coarse-grained oversimplification. A different family, the problems may be shifted differently, start earlier. But this is the problem here. Down here, the problem really is the detection. Best alignment, you would use dynamic programming, cluster type of series. Then you would, in fact, also use structural alignment of, if you have several templates, you would align them first, and then sort of see how your final model fits to this template, or the multiple structure alignment. You could use threading-like and profile. Now, in terms of accuracy, where are models? So let's just 1 angstrom resolution. That's a paper from David Baker and Andre Schally, published in Science 13 years ago. So at 1 angstrom resolution, essentially, it says 100%. So it means a very, very, very good structure. Do you have any idea what you see at 1 angstrom resolution in terms of experimental structure? So yeah?
Pretty much everything. You could, at 1 angstrom, you could probably even detect a hydrogen.
Do you see an aromatic ring?
Yes.
Yeah. Just that's the round where you begin to see that. 1.1, that ballpark, you begin to see these rings. Now 1.5, this is the realm where the applications here are a little bit overdone. Designing and improving ligands we also do on these here, but it's obvious you can do whatever you want with this. Typically, this is for the details of studying the catalytic mechanisms, designing and improving ligands, but again, it happens also below. Here, we are beginning to look at docking, which again, what binds to it, what drug binds to it. This is a ballpark into which we want to ideally come. 3.5. Any idea what that corresponds to? Is that good? Is that bad?
In terms of
what can you do with it? What is it? What comes to your mind? 3.5 is that something that is bad? Is that something that helps? If it helps, what does it help for?
Well, we were told in our practical that predicting something with 3.5 angstrom accuracy is not good. If you're 3.5 from the template...
Told you who? Who told you that?
It was during the COVID practical.
Who said that?
It was
the vast majority of people would dream. In many cases. Okay, there are models. So indeed, so I don't believe there are models in 1.5. There are models that are better than 3.5. That certainly is true. But 3.5 is in fact, many NMR structures don't reach that. So many molecules have so much motion in them that it's not that easy to reach. 3.5 is a point where you have atomic resolution. It's a resolution where you see, I mean, you certainly see secondary structure, you see contacts, you see a lot of the details. But again, so 3.5 is extremely helpful, and for many models, we would in fact like to get into that realm. Here, 4 to 8 angstrom. And now we get into also what you can reach by MD, by Vimentin type of simulations. So you have 2 helices, you have a superposition of these 2 helices. What does it tell you? Well, you find that it's 2 helices. You find somehow that you have to align them like this, which is end to end, you know, big story. You see where the kink is. And that, in fact, if you had that structure from complete wild simulation, is that trivial? That already is not trivial. So there is no intrinsically built-in breakpoint. So in the simulation, in principle, you could elongate this helix. That the helix folds back onto itself is a non-trivial point. Okay? What makes it fold back onto itself other than a breaking point that I said? In some cases we have these breaking points. So prolines are a typical example for that. But if it's not the proline breaking point, what in the simulation do you believe will drive these 2 helices together?
If they start folding at the same time and stay bonded together.
Yes, but why would they stay bonded?
Yeah, hydrogen bonds.
How strong are hydrogen bonds? Are they strong?
Well, they're what defines protein structure, so...
Yes, that's true, because there are so many of them. But when you see 2 helices interacting, you immediately see the hydrogen bonds can only be for one side of the helix. So it's every 4th residue. So we have relatively few, fairly weak contacts. while a helix is defined as a hydrogen bond for every single residue against something 4 residues away, right? Helix of 10 residues has 10 contacts, yeah?
Because of the hydrophobic effect.
The hydrophobic effect could be very, very strong. So again, if I don't put this in isolation, but if I have it in a solvent, then you would immediately see reasons why that could happen. And the hydrophobic effect, the phase of the helix, could be an immediate candidate for explaining that. And that typically, if you saw that in an MD simulation, most likely that would be the explanation. You're 100% right on that, yeah?
That's my bridge.
That's a great idea. So now, assume we had a cysteine bridge here. Is a cysteine bridge strong? Anybody else? I see a movement. I assume that means yes, right?
What defines strong? You know?
Well, it is a
No, it's fine. It's fine. It's fine. It's fine. It is strong. Now, my question is a different one. You're right on that. So you know, we and this guy, we have different body movements for yes and no. I'm not sure whether you know that. So he says ne, and you know. So it's either yes or no, and you know which part of it is, right?
But ne means yes in Greek.
Yeah, exactly. It's just the other way around. This is why with your mouth I was not sure what we were. Anyway, so my question now is the following: a cysteine bridge is really strong. Much stronger than hydrophobic. But is it strong enough to bring them together? So they are like this.
It's probably only strong enough to keep them together, right?
Yes, exactly. So you don't, and there Nico is right. So if they coincidentally, so cysteine bridges came coincidentally together, then it would keep them. This would not drive folding. Hydrophobic effect would drive folding. Okay. Okay, so that's the landscape. And again, whoever tells you that 3.5 is bad may have a point, but for many models, we would very much hope that we can reach that. Now the discussion becomes what is random. Here we have examples of mistakes of 4 to 8. Typically, some people would argue 8 is random. In some sense, you can make 2 proteins always for some subset. Manfred Zippel argues that way, fit at 8 angstrom. So there we clearly don't know. Here we clearly have useful realities. Let's talk about state-of-the-art methods. Do you know the state-of-the-art methods in the field? Out.
Modeler would be one.
Modeler would be one. Any other?
TM align.
Okay.
I don't know if it's state of the art, but there's, I think, Swiss model.
Okay. Swiss model, anything else you have heard of? Not state of the art, TM align, Swiss model, modeler. Anything else you have heard of? So let's talk about modeler. André Chaly at UCSF. André Chaly studied or did his PhD with Tom Blundell at Birkbeck, postdoc at Harvard with Martin Karplas. At Rockefeller University, he started as a professor and then went on to UCSF, San Francisco. Over 400 publications, extremely high age index, by all means a shaper and shaker. And one of the main, the piece of work that he built upon and that got big was Modeler. As I said, done with Tom Blundell originally. Another person on Modeler, or making Modeler big, was Marc Marty-Renom in Barcelona now. Modeler is a website, and Modeler can be downloaded as a program. Basically, the idea is similar to the ones that I explained before. So you find the structure, you align, you build a model, you evaluate the model, and then you sort of have different options, whether you find another template, or you just realign the template, or you just say I don't send you anything, and then in the end you sort of send it out. So let's begin with the constraint satisfaction. So you, by having a template alignment, or template target alignment, you imply a structure. imply a structure. Let's just repeat what you physically do. You have an alignment. According to this alignment, you have, for your sequence, a corresponding three-dimensional position for the other one. These three-dimensional positions you just take, copy, and then you assess. And so the first thing that you see is you imply distances. Now, these distances have to be reasonable. How could you check whether sort of the distances that you imply in your model are right?
Well, first of all, you know that you have to bridge the gaps. So, and the C beta atoms have to be at like some 3 or 4.
No, but that is, you're simply talking essentially now about the chain, right? No, I'm talking about these sort of non-local connections here, these dashed lines.
You can check whether the side chains are long enough or too short to satisfy them.
Exactly. So you can essentially look at distributions. So shown here, C alpha. It's not side chains, but is a similar story. So can we, in fact, looking at the frequency that we observe in the database, can we match the distribution that I have to the frequency in the database? So essentially that typically is called a potential, right? Distance potential. And I can apply that, and I can, in fact, find the model. If I have several models that I produce, slightly altering the alignment, I can find the model with the highest probability in terms of its three-dimensional distance match. Okay? Is there anything else I could do at that point? So I have many different models and I assess them according to the degree to which the distance maps to something that I see in the database.
I could apply another potential. I could assess them.
Just the same idea. Yes, you can now. In fact, we are also beginning to see why this business is so complicated. So you can freeze the potential and just try many different alignment techniques, or you can freeze the alignment technique and try many different potentials. And most likely what you will find is that in some situations this potential is better, in some situations that, and you can sort of begin to sort of come up with an empirical list of things that you're going to sample in the future. But I mean, in terms of an idea, is there any other idea that you could employ at this point to reassess the alignment? We'll get back to that. So this in itself, in fact, is now a procedure that is no longer trivial. It's not only that you have to create a lot of alternative alignments, but you in fact have to assess them right, and that is certainly a CPU issue. Now, essentially, ultimately what you want here you enforce is fit. Now you get into an objective function that is no longer that trivial. In the alignment, the objective function is percentage sequence identity. Now we are sort of looking at other aspects that pertain, in this particular case, to distance potentials. Or to which degree does it fit into molecular dynamics? Is molecular dynamics satisfied? Or Langevin dynamics, is that satisfied? And I actually don't know how it's said in English. Then the other issue is that so far, for all of these dynamics, I assume that the protein is a rigid body, which we know is not true. How can I change that? Well, I can introduce temperature, right? And the temperature allows me to move around. But now I'm moving around in a space where I have a model. So I know I have to do that, but I don't know how, right? So getting rid of the rigid body idea here is something that we would very much hope to be able to do, but in the space of a model it's not that easy. We do a minimization, or they do a minimization, and ultimately get back to a particular solution. Now the particular solution is the solution that you have for one template. increasingly often, I said that almost half of PDB is in fact 95%. So if you find one, the probability is pretty much non-zero that you find another one. So you're going to do that for another case. And then, in fact, explore random fluctuations, see that you can avoid local minima. Now, the typical errors that you get into are sidechain packing, misalignment, and wrong template. Let's begin here. How can you correct for that mistake in the template or in the modeling procedure?
I guess you hope that it falls through with your potentials. So if you get them.
Now, assume you have a wrong template. Can you correct for that mistake? End of story. Bye bye birdie. All that you hope, and I believe that's the question you answered. All that you hope is that you hope that you realize. Okay? That brings me to another issue. Molecular dynamics. David Shaw's Anton can fold V Menton. Let's assume that now I started from an all alpha structure. So I take a protein that is all alpha, and I put it into a fold that is all beta. And I run molecular dynamics. Will that see that we are totally wrong? It should, right?
So it should form helices again.
Exactly.
Yeah.
Does it?
Does it depend on how we run Anton?
No, that has nothing to do with Anton, and that problem is long before Anton. Or that issue has come up long before Anton. No, it doesn't.
Probably it does not.
No, it's totally happy. It's totally happy and will optimize. Or will spend your CPU, whatever you give it. But it will not recognize that as completely different from having started with the right alpha helical solution. It will fold around in the minimum, but it does not see, it doesn't have an entropy function. That is in fact why we need this sort of the zipper type of potentials, the overall view, that on the molecular level of the molecular dynamics view is not available. But this is not what I meant here in this point. What I meant is you cannot escape from the wrong template. What about the misalignment? Can you correct for that mistake?
Only if it creates classes with the rest of your alignment.
What do you mean by classes?
So, at the end of each iteration, we check our model. We check for steric inhibition. We check for this. If what we've misaligned classes with something else, then we have to reassess our alignment.
But that is again, yes, that's a good point. But you're saying the same thing as here. So I hope that my model will tell me whether that is the case. My question here is, can I correct for it in a modeling procedure?
Then I have to say no.
So in this case, I'm sorry, I'm setting you up. I'm just enjoying that, Nico. In this particular case, some of the mistakes you could correct. So you could imagine that you, in fact, have a register shift. You have the helix one loop further. And you could imagine that you have enough temperature to, in fact, overcome some local minimum and you move the helix. That could happen. Unlikely. Most likely your answer is right, but in some cases that at least you could possibly happen. So this you clearly cannot handle on the level of the modeling. That in some lucky cases you could. Sidechain packing clashes. Can you handle those on the level of the modeling? So I have two. I'm beginning. So this answer is beginning to dominate. Yes, indeed, you can. So that's exactly what the modeling is good for. That's again exactly what the optimization procedure. But still, back to the issue that I had before, refinement in the core of a protein is very difficult. You cannot always avoid all clashes. Right? It's a multi-level problem. There is a score that modeler puts out that is unique to modeler, and that contains essentially, at the end of the day, its level of measuring the degree to which it's good depends on the potentials it uses, and that score is the background to its success. Now let's get to another level, the loop. So these two here clearly also differ in these loops that they have. Let's just look at an example here from Yazara. EGF, the growth factor, epidermal growth factor in human, is in blue. The Spitz is the growth, the same thing in fly with a different name, just to make computational biologists happy. You see that in this particular case we have fairly easily bridgeable loops. I mean, come on, this is what we live on, that we recognize these name similarities. That's what biology lives on. Anyway, but let's assume you have an example where in fact in your template this connection is just very, very short, and in your target it's a very long one. So what can you do now in a case like that?
You search for...
You don't find one, is what I'm saying.
Oh.
Oh, wait a minute. I'm not sure. Let me. I was too fast, Nico. Can you say it more clearly what you wanted to say? I believe you did.
For loop modeling.
Yeah. So you all guys know. Everybody knows that. I'm finding all loops that somehow may fit to this type of constraint. That's one way in which we can model loops. Is there another way in which we can model loops? No, somebody else. Why do you know loop modeling and everybody else doesn't? Because that is part...
Yeah, to program the stuff.
Ah. And you remember what you programmed? Good.
Well, yes. They were...
Enjoy while that lasts. Okay, so what else could we do? We've talked about it.
Maybe the sheet is shorter.
It is.
The loop then is...
That in fact is a maybe that is unfortunately very, very often true. But that makes it more complicated. Because in some sense, because that means you have to sort of doubt how far you got to that point. And I had assumed here we don't doubt what we have done so far. Our doubts only come for the future, not for the past. We are fine, it's just what we're going to be is unclear. So yes, you're right. That may often be the problem. Hi.
I need to make an announcement after the lecture.
But it's not now, is it? Am I? Oh, I'm.
Yes, you have 15 minutes.
Oh, good. So the first time that I was too slow. We should celebrate that. I should celebrate it. So how can I find the connection for this long loop? Assume I don't find it in the database, and assume that I don't want to change the constraints that I have, so they begin and end point. What can I do? No iPhone movements. No, you programmed it, so you shouldn't. Essentially, this is what I can do, right? So Anton may not work for very long proteins, but it should work for a loop. Let's say 10 residues or 5 residues. So molecular dynamics should work because it's very confined.
How about fragment assembly?
But I think
the fragment assembly is the same. You look at loops, right? A database of loops. This is the same thing.
But
yes?
where do you get the amino acids for the loops from?
Oh, that is your target. Remember, you're beginning to model with a protein for which you want to predict the structure. So my assumption here was that the template, so the known 3-dimensional structure, is this, and doesn't have the loop. But your target, the one you want to model, your antibody, has the.
Okay.
I'm sorry. So then I was not right. How well do we do for these kind of loops? How long can a loop be, and we can still do it?
30.
No. So unfortunately not, because in the Anton 30 we have regular signal structure to drive us. We have the event of the global collapse, we have hydrophobic effects, we have all kinds of things. Here we have a darn loop that can dangle around in the world, and here we make way more mistakes. So that's the loop length versus the modeling accuracy. It's two slightly different views of it, but essentially you immediately see that we clearly cannot go to 30. Okay, 10 is long, yes?
But aren't the loops actually, most of the time, they can move, and if you then, yeah, how is this measured? Like, because if you just have one picture, then maybe you model it wrong with the picture, but it's moving, so maybe you got another picture, right?
Yeah, somehow I cannot do that. But I'm beginning, maybe you sort of see my point. So these two things, they both move around, but they look very different, right? So this is one loop here, and this is the other loop. You admit that they look very different?
Yeah.
So you could, no matter...
You could stretch this.
But movement of a loop does not necessarily mean this. Movement of a loop may just be this.
Yeah.
In fact, the loop may completely be confined in that direction. It may be confined not in that direction, so you may be totally right. But it may be, loops mostly somehow are confined. They mostly, you have to see, let's get back to an example of a structure. The loop cannot dangle back against the helix, or may not be able to, due to some electrostatic interactions. In this case, the way I show it, there is a tiny bit of secondary structure against a long loop. It's sort of an incorrect statement. Usually you're sitting in a very dense environment where you cannot move a loop into all directions. Okay, given the time, I'm going to stop here. So